What are vaccines?
A vaccine is a pathogen itself, or it's any component which we normally give it, inject into our body, and we try to induce an immune response against that. Vaccines are basically trying to imitate a real infection. That whenever an infectious agent invades our body, our immune system gets activated. Innate immune response, which is the first line of defense, that gets activated, and that leads to the activation of more specific antigen specific immune response, which is called the adaptive immune response. So T cells and B cells, they carry antigen specific receptors on their cells, and whenever a pathogen comes in our body, the cells of innate immune response, they are the phagocytic cells like macrophages or dendritic cells, neutrophils. They basically engulf the pathogen. They try to kill. So our body basically carries that memory, and the immune system, these cells normally go into resting state, but they carry the memory of that pathogen. And nowadays there are new technologies of DNA type vaccines or mRNA vaccines. You have heard about mRNA vaccine.
In general, how many different types of vaccines are available in the market today?
Well, there are about 6 or 7 different types of technologies how we can make vaccine. We know that all information about all the proteins which a pathogen or any cell is expressing is inscribed in the DNA. And DNA makes mRNA. Before the gene is trying to express itself, there is a, from DNA to RNA is the transcription. So that mRNA is further transmitted into a protein. But this mRNA nowadays can directly be used as a vaccine. There are certain kinds of myths also, or you can say misinformation being generated and circulated in the social media. So they all leads to certain kind of vaccine hesitancy. There are certain political, issues. Some political parties, they think that we have to oppose this, and their leaders say don't do it, because you can think about, if SARS-CoV-2 there were no vaccines, then our bodies, they generate immunity, but then to generate a certain level of immunity among the population, has to be at least 70 to 80% of population should have got exposed to those population which are not vaccinated or not exposed. They also don't get the virus, or they get a virus which has become weaker, because virus itself become weaker when it multiplies in a host which has a very strong immunity against.
Hello everyone, welcome to today's episode. Today we are going to discuss about a very important aspect of public health that has saved millions and millions of lives. From smallpox to COVID-19 to HMPV, vaccines have played an important role in preventing the spread of diseases and giving herd immunity. So in this episode, we will try to understand and deep dive into the world of vaccines, exploring how they work, their history, and the science behind their development. We will also discuss about the common misconception and concerns surrounding the vaccines and examine the latest research and advancements in vaccine technology. If you have not forgotten, very soon in our history, when COVID-19 was there back in 2020, India was literally in the forefront in vaccine development. So, understanding vaccine, understanding how it works and why it is so important, how vaccines are giving you extra protection, and especially when newer COVID viruses or different variants of viruses are actually coming in every second year. So in this scenario, when you have to be up and ready, let's gear up to understand and explore the world of vaccines, uncover the facts, the myths, and the everything in between. But before we start today's episode, please subscribe to our channel, One subscription can immensely motivate me and my team, and I promise you next time I'll give more effort in giving some quality content in the form of podcasts. Today our guest is Professor Sunil K. Arora. He is an eminent professor in the field of immunology. We already had one episode with him where we tried to understand how our immune system works and how our body actually fight against all these pathogens. Today we will try to understand about the vaccines and get more detailed insight into it. So next time when a doctor says vaccines, please refer to this podcast, listen till the end and understand why vaccines are very important for you and your family. So let's start today's episode. Welcome to the Somnath show once again. I think the previous episode which we did with you on immunity, I have received a tremendous amount of positive feedbacks and responses from the audience. And on that basis, I am again requesting you, sir. I'm really thankful that you have agreed to again come to this platform and give your valuable time. Sir, we discussed about immunity. We tried to understand point by point how human immunity works, what are the different types of immune responses we are having it, and the repercussions along with it. So in that line, there is one common topic which people are still loves to understand, and I think they should always understand, because vaccines are a certain things which has become an integral part of our life. A child is born and then the list of vaccines starts after the birth itself. So we see it has become an integral part of it. But somehow to a common person, the concept of vaccination, how it is being manufactured, what is the purpose, how it actually interferes with the immune response of an human system is still something which is not very clear, which I believe. So today we will try to learn from you, sir, what is actually vaccination doing in our body and how it is helping us to provide an extra shield of protection against various diseases. So on that note, sir, first of all, what are vaccines to you in very simple terms?
Thank you, Somnath, for once again inviting me. It's a very nice topic you have started. See, vaccines are basically trying to imitate a real infection. We can take example of any infectious agent. You take example of SARS-CoV-2 because that is the recent one which everyone has encountered and most of us have encountered. And that is the time people started talking about what is a vaccine, how it is protective, and what are different kinds of myths about vaccine. So basically, if we talk scientifically, a vaccine is a pathogen itself, or it's any component which we normally inject into our body, and we try to induce an immune response against that. As we have already talked about immunity, that whenever an infectious agent invades our body, our immune system gets activated. And with that, in that terms, we say that there is a innate immune response, which is the immune response, which is the first line of defense that gets activated, and that leads to the activation of more specific antigen specific immune response, which is called the adaptive immune response. So, this adaptive immune response comes in the form of some specific cells, which are called the immune cells of our body, and they are the T cells and the B cells. So, T cells and B cells, they carry antigen specific receptors on their surface. And whenever a pathogen comes in our body, the cells of innate immune response, they are the phagocytic cells like macrophages or dendritic cells, neutrophils. They basically engulf the pathogen. They try to kill it, and then there are certain chemicals which are produced inside these phagocytic cells, which degrade the pathogen into smaller components. And these components are then displayed on the surface of these cells.
So these cells are also called antigen presenting cells. So these cells, they present the antigen to the cells of adaptive immune response, which are the T cells. So T cells which have got a specific receptor for that antigen, they will interact with these cells, and then in that way, these cells, the T cells, get activated. Activation means they clonally expand. So that particular cell which carries that specific receptor, there is a clonal expansion. So there are lot many cells or progeny of that cells they form, and they all start producing many cytokines, and they also interact with another antigen specific cell which is a B cell that also carries a antigen specific receptor on its surface. So then B cell also clonally expand, and the B cells, they start producing specific antibodies. Now, since T cells and B cells are antigen specific, they like act to clear the infectious agent. But then after clonal expansion, once the pathogen is cleared, the number retracts. So it goes down to a normal level. But in that process, what happens is there are some cells, these antigen progenic cells, they become memory cells. So they memorize the pathogen. They have seen this pathogen. So our body basically carries that memory. And the immune system, these cells normally go into resting state, but they carry the memory of that pathogen. So that memory is important for immunity against that particular pathogen whenever the body is exposed to that pathogen subsequently in future. That is the basic funda behind vaccine also. So what we do is we try to generate a vaccine in the form of a pathogen which may be killed or attenuated, or we try to identify a specific component of that pathogen so that that can induce a specific immunity, and then we try to generate memory cells in our body, and those memory cells, they protect the individual in future for future exposure to that pathogen. So that is the fundamental basic idea behind a vaccine. So we try to generate immune response against the pathogen by using that pathogen or its component, and we try to generate that immunity against the pathogen for future. So these are all, that's the basic fundamental idea behind vaccine. So in that way, we have many different kinds of methods to either attenuate the virus or the pathogen. We make it more avirulent. It is live attenuated vaccine, or we can kill the pathogen by heat inactivation, or we can generate a recombinant vaccine also. Subunit vaccines. Subunit vaccines are like we can try to identify a protein component or a carbohydrate component or the nuclear component, whichever is more immunogenic, and that will carry the message of that pathogen to our body, and our body generates immune response. And nowadays there are new technologies of DNA type vaccines or mRNA vaccines. You have heard about mRNA vaccine. So these are all various platforms how we can generate a vaccine, and we can reduce immunity against the pathogen by using these.
Okay. Sir, I think you have also somehow mentioned that different types of vaccines, like mRNA type of vaccines. So, in general, how many different types of vaccines are available in the market today?
Well, there are about 6 or 7 different types of technologies how we can make vaccine. I mentioned that you can have live attenuated vaccine. In that case, we have the pathogen. We try to make it avirulent so that it can keep replicating, but it will not cause the disease. So that is attenuated, live attenuated vaccine. As I told you that it has to be the pathogen itself, right? But in a weaker form. Second can be inactivated. We can inactivate or kill the pathogen by heating or some chemicals. That is called heat inactivated or kind of vaccine, where we have the hepatitis B vaccine, flu vaccine, polio vaccine, rabies, they are all made by this technology. Then we can have subunit vaccine. So I told you that this pathogen has got many components, proteins, carbohydrate, nucleic acid. There are different proteins. So there is a lot of background work had to be done to identify some of the components of the pathogen which can induce a very specific immune response. So instead of giving the whole pathogen, we try to isolate that protein and then we can give it as a vaccine. So there are many different kinds of vaccines which are made by this technology that is based on subunit vaccine. And we can generate that gene by either isolating from the pathogen or we can express it by recombinant DNA technology. You can clone the gene of that particular protein and express it in bacteria and then make a huge amount of protein, and that protein is used to immunize the human beings, and that induces the immune response. Then you have the mRNA vaccine, and then you have mRNA vaccine is basically what happens is by our background science biology knowledge, we know that all information about all the proteins which a pathogen or any cell is expressing is inscribed in the DNA. And DNA makes mRNA before the gene is trying to express itself. There is a from DNA to RNA is the transcription. We call it transcription. And then each mRNA carries the information for one particular protein, right? HCV. So that mRNA is further translated into a protein. But this mRNA nowadays can directly be used as a vaccine because that once injected intradermally or subcutaneously or maybe intramuscular, so it is picked up by antigen presenting cells very quickly and then that remains extra in the cytoplasm and the protein making machinery of the cell expresses that mRNA into a protein. Suppose the mRNA vaccine for COVID-19, there the mRNA for spike protein was injected, right? And this mRNA will get expressed inside the cell and the body will start producing the spike protein of SARS-CoV-2. And thus, it's a spike protein of SARS-CoV-2, so it is basically taken as a foreign antigen and the immune response is generated against that. And the body started making antibodies and cells are activated against that in a similar way as it would have infected us, right? So the mRNA produces a immune response which is against that pathogen. So that is basically mRNA vaccine. And then there are new platforms like DNA vaccine where you clone the gene of this spike protein in a plasmid and that plasmid is used as a plasmid is basically a circular DNA. It can be a virus, a viral genome, or it can be any plasmid genome, a plasmid DNA. So the gene which is of the pathogen, the spike protein, is cloned in that. And then that DNA is used as a vaccine. So the beauty about DNA vaccine is that it is more stable, much more stable than mRNA vaccine. It doesn't require that kind of conditions of sub-zero temperature for storage and transportation because mRNA is very unstable basically. It is very fragile molecule. It gets degraded very quickly. So it has to be handled very precisely at very low temperatures. It has to be stored or it can be transported also at very low temperatures. But DNA vaccines are not like that. They are much more stable. They can be stable at room temperature also. They can be transported at that level also. They don't degrade that fast. And then they also carry some part of bacterial DNA because these plasmids are basically from bacterial origin. And these bacterial DNA, these plasmid DNA, they normally have specific motifs which are called CPG motifs. And that CPG motif basically activates our antigen presenting cells. because antigen presenting cells, as I told you in the last episode, that whenever these phagocytic cells like macrophages or dendritic cells, if they look at a pathogen through their pathogen recognition receptors, which are called the PRRs, they recognize certain pathogen associated molecular patterns, which are the components of pathogen on their surface. So as soon as they do that, this interaction leads to activation of these macrophages or dendritic cells to express certain proteins on their surface, which are called the co-stimulatory molecules. And these co-stimulatory molecules, they interact with the T cell, because T cell expresses many molecules besides their T cell receptor. And that interaction basically sends a second signal for activation of T cell. So T cells require always more than one signal to get activated, optimally activated. And these DNA motifs, which I am talking about, the bacterial origin, they are basically pathogen associated molecular patterns. So they act as sort of boosting the immune response, in sense a better immune response is generated because the T cell activation takes place. So these are all different kinds of vaccines which are generated depending on the pathogen. Basically, a scientist, they decide which particular technology will be best for a particular pathogen, so that an appropriate kind of immune response is generated, and that leads to the formation of memory cells, and that memory cells will protect you in future for different.
Right. Sir, I have maybe very, it might sound silly, but the question is for COVID, during COVID times, we all knew how, India and other countries were literally struggling to come up with a vaccine at the earliest. So there was research and development team was working on war footing, and still, it took certain time. So is it because the strain of SARS-CoV-2, we were not able to sequence it and develop a vaccine against it? Is it the reason, or what was the reason behind all this hardship and the time taken to come up with a vaccine?
See, as I told you, that lot of work has to go into the laboratory, from the laboratory to identify the pathogen, or to identify the specific component of the pathogen which can be important from the point of view of generation of immunity. In case of SARS-CoV-2, it was basically required to know how SARS-CoV-2 enters into our body, how it infects the cells, and how it generates a kind of immune response which may be protective against that. So there are certain countries like China and America, they did very quick work. They did the sequencing of the SARS-CoV-2 genome, and they understood the basic biology behind the infection, that the spike protein which is expressed on the surface of SARS-CoV-2 is important from the sense because this interacts with a specific receptor on the cells in our lungs, and these specific receptors were ACE2 receptor. So spike protein, unless it interacts with ACE2 receptor, it will not enter into the cells. So when that whole sequence was done and they knew about the sequence, they started making vaccines. So all those countries which have that technology, or the development of science and technology, or the platforms ready, they were able to make vaccines very quickly. While all those countries which do not have that type of technology, and they could not have access to that technology or platform, could not make vaccines, right? So making a vaccine takes lot of background work, lot of trials, lot of regulatory issues are there. So all these things were basically behind making a vaccine. But since it was an emergency that time, so all those vaccines which were developed, or the technologies which were developed initially by the countries like China, America, or maybe India also interacted with European countries like AstraZeneca. They collaborated with them, and they got that technology. And India also developed its own technology in the Serum Institute of India, where they generated that heat and it inactivated viral vaccine, the Covishield, right? So all these countries, they started making vaccine, and they got the regulatory authority, WHO's permission to use it in the emergency. But those countries which could not make vaccine, they had to import it. So they had to really depend on the vaccine to be given by these countries. And all these countries, they started making vaccines doses for their own population. And then whenever it was surplus, they will try to store it for the future use also, because nobody knew how many booster injections it will require to create a immunity so that the pandemic can be stopped. So that was the time when a lot of disparity, you can say, in distribution of vaccines was there. But then slowly it settled down, because then the knowledge was created, because side by side people were trying to do lot of, there was no animal model to do all these kind of studies. So people generated animal model, they started doing, so much of research was being done, and a lot of money actually by the national funding agencies were put in for generation of vaccines against SARS-CoV-2. So that's how slowly the situation improved. But initially, there was a lot of turmoil, and as you have said, that many countries, they could not have access, and there was a lot of disparity in that.
Sir, in this line only, I have one. I mean, I'm not clear in that. So I have one more question in this same context. If I have prepared a vaccine based on mRNA technology,
Uh,
now that is basically the response or constructed with the genetic makeup of an individual to some extent. Now, from two cohort, this genetic makeup also will vary. So the same vaccine will be as effective in in the one population, and it is also expected to be equally effective in another population.
It's a very good question. Basically, that difference remains. But when we are generating vaccine, we try to take care of that so that it can be. So every antigen, the antigen presenting cell, they use this MHC molecule to display that antigen on the surface. So that's where you are talking about the genetic difference. Genetic difference is basically the HLA difference. So HLA molecules, they are used by the antigen presenting cells to bind the antigen and display it on the surface so that the T cells can look at it. So there is a binding of pathogen molecule to the HLA, and that conjugated component is displayed out. So ethnicity or various cohorts, they all have differences in HLA. So then there is bound to be some difference in generation of immune response. But when they try to decide what particular epitope or component of this pathogen is to be used as vaccine, they look at that most commonly expressed HLA type should be able to bind to that antigen.
Right.
On the other way, I will say in the reverse, that that component should be able to bind with the HLA of different ethnicities or religions. So they try to look at an array of HLA types, and then they try to see that its binding should be with everyone sufficient so that it is displayed on the surface. So unless it is displayed, it cannot be seen by T cells. So that is a very important step, but that is basically too scientific for the general public to understand. But I'm sure those who understand what is HLA, what is the human leukocyte antigen, these are the major histocompatibility complex antigen type 1 and 2. They are the one which bind to the antigen and display it on the surface, and then they are seen by the T cells, and the T cells get activated. So there are some differences, and that is because of HLA, and the response is a little bit different. But normally, most of the vaccines are for global use, because they bind to a diverse array of HLA types.
No, nicely explained, sir, because I always used to think that if you are uh, preparing a vaccine, then that vaccine, especially in the scenario of India, where we see a lot of genetic diversity are there across the country. So,
Yeah.
one vaccine, if it is developed, maybe for SARS-CoV-2 also, so that should be able to give the protection against this virus across the country in material of different genetic makeup which they are coming from or ethnicity they are coming from. So that was my doubt, and thank you. You have explained it very nicely. So I think the people who will be listening this podcast, they will also get a fair idea that how actually vaccines are being this. So I would say rather there is a lot of hardship behind manufacturing vaccines and coming out. We had been impatient at times when we saw the first wave of COVID, and then we started saying all things. But yes, there are a lot of team who had been working in manufacturing vaccines. Now, sir, as I said, journey from right after birth, the vaccination starts, and then doctor, pediatrician gives you a chart of vaccines which has to be given on certain intervals. So, my first question is, how do they decide that every 6 months one vaccine has to be given, or every after 2 years a particular vaccine has to be given to a child? How that chart is being? Is there any roadmap to it? How it is being done?
It's also very good question because that's all a background work which is done for while developing a vaccine, and it is actually different for all different vaccines. As I told you that once we expose our body using a vaccine can be different type, so it has to be injected, and then the cells get activated and then the memory cells are generated. But the amount of memory cells which are generated, how long they survive, normally they are lived memory cells. And for some pathogens like measles, smallpox, chickenpox, these viruses, they induce certain kind of immunity, which is lifelong, nearly lifelong. Normally, a person who is exposed to these viruses, they don't get a second bout with the same virus. However, when these kind of viruses they change and they come back as a mutated virus in a different form, then they definitely can cause infection. But the related, because they are somewhat related, so some kind of immunity works. In the SARS-CoV-2, not all of people they got severe disease. Some people they get exposed and they were detected because they will not show any symptoms or they will have very mild symptoms. Almost 85 to 90% people, they will recover automatically. Even in the first phase of SARS-CoV-2 epidemic, you saw that, right? Only few people, 10 to 15%, they started showing symptoms which required hospitalization, and only 3 to 4% were there who ended up in ICU or unfortunately died. So that means there was certain kind of immunity in the larger population. So how that immunity was developed? That was because of exposure to related viruses. And you remember in 2003 or so, there was another epidemic came, which was SARS-CoV infection, which was Spain virus. It started from Spain and developed in some countries and then stopped. So that was a very related virus. So people who were exposed to that virus or they got vaccinated against that, they never got very severe symptoms of SARS-CoV-2 because the immunity generated and memory generated against that virus was sufficient to give some kind of protection against this also, right? So there has to be some basic idea behind how the immunity has to be generated, because there is a when we give first injection, then some kind of immune response is generated, some memory cells are there. But they go down. And that immunity stays for some time. The primary immune response is normally narrow and only few cells are generated. But if you give a second injection or there is a exposure, then the second response is quite quick and more fulminant. There is more expansion of those memory cells, and there is larger number of cells which acquire that memory. So that gives you uh, a better protection because they stays for much longer time. So it is basically different for different pathogens. So that is how they decide that a second injection, when it has to be given, up to 6 months, or maybe up to 3 months, or maybe 2 years, or 5 years, or never, right?
Yeah.
So, or never, because this chicken pox or something, if you have acquired them in the childhood or you were vaccinated, in adult you never get it. Very rare that you will get it, right? So that's how it has to be science behind it. You have to understand that part. That goes into the investigational stage when the scientists are working all the time in the laboratory just to understand that immunity against that particular pathogen, which is different for different pathogens. And that's why the vaccine boosters cannot be given at the same time. It has to be different because they are different pathogens.
Right, right.
That's how it works.
Sir, I think you mentioned about boosters also. As you know, to keep our memory cells with the memory that this is the particular type of pathogen you have to be active, we give multiple, maybe 2 years, 3 years time, so that the memory cells are active, as you said. And but what is the role of boosters then?
So I told you that booster is actually the same vaccine. It is given second time or third time,
Oh,
and they will generate a secondary response every time. It's a primary response and secondary response. And whenever the number of cells they go down, so the below a certain threshold level, so you can check it by T cells, specific antigen specific T cells, or you can check by antibodies for hepatitis B viral vaccine. So people check the level of antibodies. If that is detectable and the titer is reasonably high, you don't need a booster. But if it goes down, then you give a booster, it will be restored again. So there is a certain level of antibody level or the T cell activated level has to be maintained to maintain the immunity against that particular pathogen.
yeah.
So the boosters, they basically boost the immune response against that vaccine so that a level of immunity is maintained for longer period of time.
Sir, like the in plain language, can I say that if one same vaccine, if it is repeated next time, then it is basically a booster dose?
Yes, or even the infection, even the infection. If you have taken a vaccine and then you get infected after 6 months, the infection will also act as a booster
Oh,
because it will also boost the immunity. Ultimately, the memory cells are against the pathogen only, right? And they will get boosted and they will have the expanded memory cells and they will work. So that is called hybrid immunity. And you must have read in many papers, or you can see in the so those people who were vaccinated and somehow they got exposed, their immunity level was much better, or they were initially they had the SARS-CoV-2 infection and then they were given a vaccine, their immunity levels are much, much better because that will create a lot of good immune response generated against the pathogen.
sir, I think vaccines are something which has been like every people knows what vaccines are because they have to take vaccines in their lifetime. Now, being a part of a lifestyle for such a long time, still I see people have lot of misconception as well as certain amount of fear, or you can say, some gray areas in terms of understanding what vaccines actually do. So for an example, during COVID also, when the first vaccine was came into the market and the government was literally requesting people to go and get the vaccines, there were a lot of people who were apprehensive and they were refusing to take vaccines. So why vaccines being so prominent part of our life, still people have this apprehensions and people have these doubts of taking vaccines?
What happens is You have rightly said that vaccines have basically saved millions of lives, right? So they are basically tools to protect. They generate a kind of immunity which basically reduces the person to person transmission. So, but what happens is there are certain people who are called anti-vaccine community. They, because of religious reasons or some other faiths, their religious leaders, they tell them not to take vaccine because they don't allow their people to get an injection of something which is basically not happen to them. So vaccine is prophylactic. Prophylactic means you are not exposed to some pathogen, but you want to create a kind of immunity against that. So in anticipation. So that's why it is called prophylactic vaccine, right? So some people, they don't believe in that kind of ideology. Why should we take something when we are not exposed? So that's one thing. Second thing is some kind of apprehension of negative effect of vaccine, right? So like you rightly said, when we had these COVID vaccines coming up, the mRNA vaccine, many people thought that mRNA will go into our cell and it can actually change the DNA sequence so that our children will become some generate certain different traits because DNA carries the main message. But it is all wrong information. Yeah. Some kind of misinformation is circulated because those people who know that the mRNA can never go and bind to DNA, but it does in some cases, like HIV. So there are bad examples also. The HIV kind of virus, which is a retrovirus, it is also basically once it infects our cells, RNA goes inside our cell, which is, and this RNA converts into DNA.
Sir,
And
something is
DNA,
something is coming in your screen. Your background changed. Something happened.
oh, if I do this, then
Oh, it is some thumbs up came. Then some firecrackers came. I thought you were having New Year celebration.
okay, I'll say again. You know what you can.
I'll delete.
So what happens is, some people think that the mRNA will go and change the DNA sequence, but it doesn't happen like that for COVID, the SARS-CoV-2 vaccine, because this mRNA remains in the cytoplasm and get expressed into spike protein. I know that some viruses like retroviruses, the HIV, the RNA goes and integrates itself, but then HIV has got those enzymes which can insert that RNA into the because it reverse transcribes the RNA to cDNA, and cDNA goes into the nucleus, and there enzymes, the integrase enzyme helps it to get integrated into the human genome. But that doesn't happen for SARS-CoV-2 or the mRNA vaccine. So mRNA vaccines which are generated for SARS-CoV-2 or for Ebola or some flu virus, they never go and bind to DNA. So there are certain kinds of myths also, or you can say misinformation being generated and circulated in the social media. So they all leads to certain kind of vaccine hesitancy.
Yeah.
There are certain political issues. Some political parties, they think that we have to oppose this, and their leaders say don't do it, and they bad faith will just go and say.
Yeah.
During SARS-CoV-2 vaccination, it happened. People said, why our prime minister is saying the vaccine, so we will not do it because of political reasons, religious reasons, because of so all these kinds of things they lead to and vaccine hesitancy. Otherwise, vaccines are very important. They are protective. They actually help in so many other ways rather than just being protective. Because you can think about, if SARS-CoV-2, there were no vaccines, then our bodies, they knew they generate immunity. But then to generate a certain level of immunity among the population has to be at least 70 to 80% of population should have got exposed to the virus.
Yeah.
It takes time to generate a certain level of immunity so that the transmission from human to human is blocked. Because so many people in a community should be immune to the infection. So the virus is not transmitted to those. And that's calculated by at those population which are not vaccinated or not exposed, they also don't get the virus, or they get a virus which has become weaker, because virus itself become weaker when it multiplies in a host which has a very strong immunity base. Okay. If you have two hosts, one is very susceptible to this and doesn't have any immunity, the other one is vaccinated or has got immunity against that, then the virus which is replicating in this immunocompetent host will be weaker, like will come out, it will have lesser capacity to infect the others as compared to this. So we call it replication competent virus, and it becomes more virulent, right? So that is a basic idea of generation of herd immunity. Herd immunity is like 80 to 90% of people are immune to the infection. And that can be generated only very quickly by vaccines. But in a long run, if the infection goes on and on for many years, then you see that herd immunity is generated. So it is better to generate it much quicker than giving a mass vaccination. That will generate a kind of immunity against that.
Right.
Does this.
Right.
So it's very important.
Right. Right. No, sir, I think apprehension towards vaccines and whatever you said, they're all mostly what I understand is non-scientific reasons are there, which is actually making false alarm within the populations. But otherwise, scientifically, if I say vaccines are important, people should go and get their vaccinations done, and proper vaccination can save them from a lot of much before and well ahead in time, right?
And so SARS-CoV-2 apprehensions came because vaccines were given emergency use approvals. Emergency use approvals were given when the clinical trials were done, but some of the data was not available. So that was not analyzed. So the real data comes from the mass population.
Yes.
And for the long-term effects of a vaccine, you have to wait for many years to see those long-term effects. So that could not be done in this. That's why there was some
Yeah.
apprehension in scientific circles, because many scientists they believed that a vaccine has to be seen for its side effects for longer period of time.
Right.
Otherwise, it may generate certain kind of effects. So those side effects were not known because some effects were not known. So I don't blame people for some time. There were some scientists who basically talked on scientific basis also. But that was an emergency, situation. That's why those apprehensions were there, which are mostly now removed. Most of people have reason, because the long-term effects have already been seen now. Because the real test, which is called of a vaccine, comes only when it is given into the masses, not just a small population in the form of a clinical trial, which is phase 1, 2, and 3. It's still 1,000 people or maybe a few thousand people in clinical trials, phase 3. But then you have to give it to millions before you see that rare side effects, which comes up after a long time.
Sir, in line, maybe what is the time period for clinical trials?
There are basically this is a set protocol: phase 1 clinical, 2 clinical, 3 clinical trial, and then the long term effects, right? That is phase 4. So every phase has got like the time period will depend on the cycle of infection and how early we can see the impact of that. Like, one is a short one with a smaller number of people. Phase 2 is a little larger, but then you have to see the side effects also. Phase 3 is much larger population. And so taken together, it may take maybe 10 years or 15 years for a vaccine to complete all these clinical trials.
Okay.
Go for some pathogens can be shorter also, but the long-term effects need to be seen for at least 5 years.
Okay.
So that's why people were apprehensive for SARS-CoV-2, because within a year you want to give a vaccine which are to mass.
So it was basically, to uncharted lands. We were not knowing the repercussions in long-term effect,
Right.
and that might have uncertainties. But yes, sir, we cannot deny the side effects of vaccines. So in generally, what can be the side effects of a vaccine one can have?
That again depends on what kind of vaccine you are using against which pathogen.
Okay.
Of the vaccines which are generated against using a technology which is kind of innovative vaccine. So there you have the live pathogen, right? Live pathogen attenuation can reverse also, and that can the pathogen also. And these live attenuated vaccines are not recommended for population for those people who are vulnerable, like females or elderly people, or those who have certain comorbid conditions. So because their immune response is a little bit compromised. Whereas other vaccines, kinds of vaccines now, recombinant vaccines and all, they are very good and they do elimination.
Okay. Sir, I think I had lot of questions on vaccines, and sir, I on this context, side effects, as you said, will vary from person to person, right? And the manifestation of side effect, one person cannot will be the same, or it need not be same. It can vary from different people can have two different manifestations of side effects.
Yes, two different people can have different side effects depending on their own vaccine inside will produce a immune response, generate an immune response. Then the vaccine may carry certain kind of the adjuvants, and adjuvants, they basically boost the immune response. Basically, they facilitate the generation of immune response. So in some people, response can be different. You may generate certain kind of effects.
Yeah.
So side effects, people start talking at the site of injection, fever, or kind of body aches. And then long-term side effects can be in the form of are saying that there are increase in incidence of heart attacks because of coagulation problems. Those kind of effects can be different for different vaccines. You cannot generalize them for all vaccines.
So I got a very clear picture, because even I had certain doubts in my mind that how these vaccines in totality work in our body, and whether the vaccines are always safe or there are any to taking vaccines. But thank you, sir, for giving a very elaborate, very lucid explanation of how vaccines work in our body, how it interferes with our immune system, and then one should not be very much apprehensive in taking vaccines from next time. If anything is there in your, I think all the viewers should definitely go for vaccines and complete their vaccination course. With that note,
Yeah, one thing I will like to say in the end, sorry for interruption, that once the vaccine is released, it is approved by regulatory authority. CHO is the World Regulatory Authority, but in India we have the, the Central Drug Organization, CDSCO it is called. So this a national regulatory authority.
yeah.
They look at the whole data, they look at the protection, they look at the side effect, they look at the mode of, route of administration, how many boosters have to be given, and what large population can be covered with that vaccine. So all these aspects are seen before a approval is given for the mass vaccination.
Yeah.
Once that is given under very tight protocol. So once that is given, so people should have very less apprehension. They should not release the windshield. So that we should believe that the regulatory authorities are working the scene, and they give approval to only very safe vaccines, and they give clear indications that this vaccine can be used on vulnerable population or not, and to some extent to the children or adults or pregnant females. or elderly. These are all which are that are released along with the vaccine. So I think we should not have apprehensions once a vaccine comes in the market. So it is actually very safe because of these regulatory.
Yeah. Thank you, sir. Putting it in a very simple, subtle, and in a way you have conveyed a very straight message that vaccinations is important. You should not be shying away from taking your vaccines on time. Talk to your doctors, get to know what all vaccines are there for you, suitable for you and your family's protection. Please, should go and get all the vaccines done. So
Yeah.
thank you, sir. This point or this questions keeps coming because vaccination is something which I said right at the beginning of the podcast. It has become a very fundamental part of our lifestyle these days. Right from the child is born, the pediatrician will give a list of vaccinations. And if somehow people think that it can do away with one or two vaccines, the repercussions might be somewhere down the line when the child grows and then sees or experiences certain type of diseases. So God forbid there should not be any such situation, but we should be well
And the
prepared.
real impact of a vaccine comes when the mass population is vaccinated. Because if they are for children, almost all children should be vaccinated. Otherwise, the virus or pathogen will keep. Hereby, the polio vaccine, they say that not even a single child should remain unvaccinated. So every child should get the vaccination. So that's how you can break the transmission chain. So you have to break it only by mass vaccination.
Yeah. And rightly said, sir, polio has been almost, I can say, it's eradicated from India, but still the polio vaccination, the polio drops are being given. So it's a very good initiative from the government of India, and without any fail, your newborn should be given a polio drops right on time, because one step at a right time can save lot of problems in future. So when we are starting a new year, I think this is a wonderful note or wonderful platform we have where sir has set up a wonderful message to all of you that you be taking care of your health, and not only take care of your health, get to know what all vaccinations are there in your chart and get yourself future ready. So with that, thank you, sir, for your fall short of words to appreciate the fact that you have given your time and your wonderful knowledge, which I think the audience would be immensely benefited out of it if they follow even slightest of it. So with that hope and with that note, thank you. And I would like to, as I said in the previous episode, if time permits, we would again connect with you, maybe on a different topic, but definitely would love to talk to you and get a in-depth insight on topics from you as much as possible, sir. Thank you.
Thank you very much for inviting. And I enjoyed talking to you once again.